Hello everybody, you know the drill. I'm going to go fast today. We're talking about exam 3 from spring 2014. First question, what enzyme or which of these reactions is part of gluconeogenesis and glycogenolysis? So glycogenolysis being the breakdown of glycogen. The answer to this one is B. So the enzyme is G6-phosphatase. Other things I'd like to ask about that are that's the one that's ER bound. So it's membrane bound to the ER. It's found mostly in the liver. And phosphatases use a water. So as always, good practice to make sure you know where all of these other reactions happen, because the next question on the next test could be about one of these guys. So the other reactions, where they happen, what the enzymes are. Which protein does not have catalytic activity? So obviously all enzymes are going to have catalytic activity. So we can cross out these popular enzymes. So it's between GLUT4 and glycogenin. If you look on slide 30 of Unit 7, you'll see glycogenin does have a couple enzymes, so it has catalytic activity. GLUT4, the glucose transporter, does not. So that's the answer. Which protein is turned off by phosphorylation? It's glycogen synthase. So 33, slide 33 shows some of the regulation of glycogen synthase and glycogen phosphorylase. So make sure to know that one really well. This molecule does not appear to stimulate. When I was doing this, anytime I saw genolysis, I would just say degradation, because that's how it was in my head, so if that helps you. So you don't mix up glycogenolysis, glycogenesis, and glycolysis, which all look pretty similar. I talk about that in the other video. So glycogen degradation in the muscle. Heart's just another way he says muscle. And the answer to that is glucose. So the purpose of breaking down glycogen is to make glucose. So if you already have it, it's not going to stimulate making more of it. Which reaction is called phosphorylasis? I'd never seen phosphorylasis used, it might have been on one of the slides, but basically lysis, so a break or a cleavage with phosphate. So phosphate doing the action. If you're going through, the only ones where a phosphate is taking off something is E. So you have glycogen here, you add a phosphate, and then you have one less glycogen and one free glucose molecule. So you broke off some of the glycogen with a phosphate. Glycogen phosphorylase is the enzyme there. So it makes up phosphorylase, like glycogen phosphorylase, and phosphatase. So phosphatases we see are using water, phosphorylases are using phosphates. Which is not correct for a fasting liver. If you're fasting, you're going to be breaking down glycogen. You're not going to be making more glycogen. So glycogenesis is the synthesis term. It's not going to happen in a fasting liver. That's a fed state activity. Which protein is not used to make a glycogen particle in the heart, muscle, from blood glucose? So not used to make a glycogen particle. So we're talking about the synthesis reactions you have memorized, and debranching is in the degradation reaction. Remember, in the synthesis you'll see things like the first step of glycolysis. You'll see how it got into the muscle, how it got into the heart by GLUT4. So remember, GLUT4, muscles and fat, GLUT2, liver. Keep those straight. They could help you on a test question here. This enzyme is used in degradation and synthesis. Like I said this a bunch before, it's phosphoglucomutase. while we're talking about that one, let's skip ahead a few. Those are the things he loves to ask about it. It's used in both reactions, and it's not regulated. So those are the main things to know about phosphoglucomutase. Which metabolite is essential for glycogen degradation in liver and muscle? Degradation done by glycogen phosphorylase, which uses phosphates to free off the individual glucose molecules. That slide 18 has a bunch of questions from it, so make sure to know that really well. Which metabolite appears to stimulate liver glycogenesis? So the same way I crossed out glycogenolysis and wrote degradation, I would always just write synthesis here. That just helped to keep it straight in my mind. So if you're making more stored glucose, glucose is the answer. If you're going to synthesize more stores, you need supplies to do so. This glycogen enzyme is known to be partially activated by AMP in the muscles. So here's 33 and 37 are two good regulation slides to know. Glycogen phosphorylase is the answer. It's partially activated by AMP like it says. I think you see that on 37. On 33 you see the other way it's activated, which is by phosphorylation. Phosphorylation is done by pKa. So pKa adds the phosphate, which activates it partially. AMP is the other partial activator. These metabolites does not appear to regulate glycogen metabolism. So in slide 37, it's the good regulation summary slide. All of these are listed at some point. Make sure to know where and what enzymes these work on. I don't think I've ever seen phosphate work in any sort of regulation, all through Biochem too. Obviously, energy charge does. Liver phosphorylase is working with the glucose, and G6P acts in there as well. Which molecule is a substrate for UDP glucose pyrophosphorylase? These are memorizing the synthesis reactions on slide 23. The substrate used by this enzyme, so remember, substrate is just a reactant, and that's UTP. Which condition typically stimulates gluconeogenesis? Remember, gluconeogenesis is a liver, so glucagon is acting only on the liver. It's reciprocally regulated to glycolysis, so if glucagon inhibits glycolysis, it's going to stimulate gluconeogenesis. So insulin would be an inhibitor of gluconeogenesis. Which vitamin is always required for gluconeogenesis? It's niacin. So 36 is a good summary slide of all the vitamins, ATP requirements, and enzymes for the different precursors to glucose. One of these liver reactions is stimulated by acetyl-CoA. It's going to be B. Pyruvate carboxylase is the enzyme that does that, which is stimulated by acetyl-CoA. So I wrote these out for a couple different questions. 38, 39, and 40 are all different ways of showing the gluconeogenesis regulation. So if you're memorizing only 3 slides, those are the 3 to do it. 36 is also the other really important one to know. Glucose precursors generated only by fat cells under normal conditions. So fat cells, it's going to be glycerol. Lactate, muscle red blood cells, proteins, I'm not sure if he was trying to trick you. That seems like kind of an obvious one. The glycerol is the backbone to triglycerides. Which of these enzymes is not vitamin dependent? Answer is aldolase. So these dehydrogenases are both niacin dependent. Alanine aminotransferase is B6 dependent. So make sure to know that. It's the first time we see B6, so he'll most likely ask about it. And PDH is 1, 2, 3, and 5. Twelve enzymes, 3 vitamins, 6 ATP equivalents to convert this molecule to glucose. So 36 is the summary slide, so you can find it on there. Alanine is the answer. Alanine is in the pyruvate family of amino acids. So I think if you look on this slide, it'll make sense of where that's coming from. But this is the format he's going to ask. He might just change the numbers around and have a different answer. So make sure to be comfortable with this style. It keeps coming up over and over again. Gene suppressed by insulin. That's pep carboxykinase. That's found on slide 38. These molecules can be converted to glucose without a carboxylation reaction and without an amino transfer reaction. So that's glycerol. Amino transfer reaction is going to take out all of these amino acids, and lactate comes in at the pyruvate level, and then the next step is a carboxylation reaction, so that's why lactate's not an answer. Next question, not a redox enzyme. Hopefully if you didn't know any of these, dehydrogenase are all redoxes. This is the different one. That's the enzyme class 6, a ligase reaction. The other thing, I don't think he asked about it in this test, but he likes to make sure you know it's biotin dependent. These molecules cannot be converted to glucose. We've seen all of these amino acids are all the lactate and glycerol. Palmitate is just a fatty acid, so that can be converted directly into energy. You wouldn't need to turn it back into glucose. A couple regulation questions. Not found in a fasting liver. This is our friend from Unit 3, the most potent activator of step 3 of glycolysis. So it would be found in a fed liver, not fasting. Inhibiting gluconeogenesis. It's AMP. So make sure to know these regulation slides really well. He's going to ask about very specific pieces from those slides. This molecule is always needed for gluconeogenesis. The answer is water. Those phosphatases that you see to reverse steps 1 and 3 of glycolysis, those phosphatases use a water. So even if you're using glycerol, which enters in at the highest point up in glycolysis into gluconeogenesis, that's still requiring some water. This is amino acids only. This is stuff entering in at pyruvate only. Same with this, is the pyruvate only. It's involved in the carboxylation reaction. Enzyme is found only inside the mitochondria. Pyruvate carboxylase. Slide 33 has a good summary of enzyme locations and where they're found. All right, I'm going to go quickly through the last unit. The pathway does not require NADPH. It's glycogen biosynthesis. Metabolizing ibuprofen is cytochrome P450. It's found on the very last slide. This enzyme does not appear to be vitamin dependent. Phosphopentose isomerase. These guys are, again, 1, 2, 3, and 5. dependent, and transketolase, he wants to make sure you know that one is B1 dependent. It's a TPP vitamin. So make sure to know that part. I think you see that on slide 14. So this is just knowing these pathways. I have them written out much more in the other video of knowing which cannot be metabolized by transaldolase. The answer to that is the xylulose 5-phosphate. So there's a lot of clinical stuff on here. Enzyme involved with favism, glucose 6-phosphate dehydrogenase. This molecule is not an oxidant. So all of these are oxidants. Glutathione is an antioxidant. We saw that in Unit 6. This describes the function of the PPP in red blood cells. So all of these other things are pretty specialized functions having to do with fat and having a mitochondria. Maintaining reduced glutathione is the job for red blood cells. All of these other things are more specialized cells that we kind of learn about mainly in the next unit, in the fatty acids unit. Which enzyme is known to metabolize organic peroxides? Peroxides, glutathione peroxidase. See that on slide 32. Reaction taking place mostly in the liver is the last one here. Metabolizing these antibiotics, or xenobiotics. Slide 24 has the PPP regulation. Very simple. Inhibiting the PPP is NADPH. That's kind of the main function, or the main reason why the PPP exists. So if you already have it, it's going to inhibit it. If you have a lot of this, that would stimulate it. Another clinical question about Heinz bodies. So you really wanted to make sure you know the details of it. The answer is C. You can see that on slide 34. The last one I couldn't find a slide reference to or an explanation. He might have mentioned it in class somewhere, but the answer on his answer key is C. There you go. Good luck with your test.